Yes. Moving swiftly on to metagenomics of ticks. So why are we interested in ticks? Well, ticks are what we call ectoparasitic arthropods. That means their parasites live outside your body and they feed exclusively on the blood of their vertebrate hosts. And the problem is, across the world, they act as vectors of both human and animal pathogens. And these pathogens include viruses, bacteria, and protozoa. And what's worse, what's more, is that not only do they transmit from one individual within a species to another, but they can actually mediate transmission of pathogens from one species to another, from one host species to another. And in particular for us, that means from an animal to a human in what we call a zoonotic infection. But we don't really understand very much about the prevalence, diversity, and the virulence potential of these tick-borne agents. And so there is a need to go out there and look in the world, in the outside world, in the wild, to see what's out there, what is actually moving around in ticks, what's present in ticks that are biting animals that associate with humans. And in fact, although this particular study is a few years old, I am just at this very moment reviewing applications for a call for proposals to do very similar work here in the UK that's been launched by the research councils, and they're very keen on understanding the potential for arthropod transmission within the UK, particularly as we've seen some of these arthropod-borne diseases getting close to us. In, for example, France, they have a dengue outbreak. And we did used to have malaria in this country, and we still have mosquitoes that are capable of transmitting malaria. So we must not be complacent. But I'm not going to talk about the UK. I'm going to take you to a far away, more exotic part of the world, which is the West Bank Palestinian territories. And so what our collaborators did, I didn't introduce them here, the three collaborators there, Abed, Omar and Suhair from Palestine, and then Anu Ravi, who used to work for me as a postdoc here in Norwich. We worked together on this project. And what they did was they went around various towns and cities within the West Bank of Palestine, and they collected these three different genera of ticks from these three different mammalian hosts, so dogs, camels, and sheep. And you can see that they collected different genera of ticks from different sites. In some sites there was only one, in some sites two. And one of them, hyaloma, is only present on the camels, and there's a particular niche for that. So one of the first things, what we did was we just got them to mash up the ticks. We sequenced the DNA en masse, and we analysed it. And one of the first, this is a table that generally illustrates what we found. So one of the cool things about using metagenomics in this context is that we could actually look at tick DNA, we could look at the host DNA, so the ticks had had a blood meal, carried blood from a host that they had recently bitten, and we could also look at what was living inside the tick in the time in terms of microbes. And one of the things that we found quite quickly was that we could see that some of the identifications of the ticks that had been done on morphological grounds didn't match what we were finding on sequence grounds. And it opened up the potential that you could identify the ticks far more easily by just sequencing them than by staring down a microscope and doing detailed comparisons with a kind of key that you hold in a book to see what the tick actually looks like. We did find the host DNA that was identifiable in a number of these. You can see we've got sheep and dog DNA in some of these samples. In some we couldn't identify it, and it's a bit hit and miss. Depends on how heavy the blood meal was and how much of it remains at the time that one actually takes a tick and samples it. But the fact that we could do that I think was still pretty cool, that you could actually identify the host without too much difficulty. And then we found a number of key microbes in the taxonomic profiles we got there, and I'm going to give you some more details on those as we go on. What we actually did, even though, as you probably heard from Chris's comments, I'm not a great fan of 16S, but in this study they did do 16S first, and then we homed in on the ones that looked interesting using shotgun metagenomics. And the shotgun metagenomics gave us a much higher resolution view of what was going on. So when we did that, we found that 2 of the sheep ticks yielded an abundance of reads from the genus Rickettsia. So this is known to be a human pathogen. It's an strange kind of intracellular bacterium. It has to live inside host cells. And we got 2 assemblies out. One assembly was very similar to an isolate from a species called Rickettsia massillii, which then if you know Latin means that comes from Marseille in France, so right up the other end of the Mediterranean. But there was extensive synteny, and there was only a relatively small number of SNPs across the genome, suggesting that these tick-borne agents can actually travel over quite large distances, presumably through the movement of livestock, probably that rather than humans, but it's not certain. Now we know that this Rickettsia causes a disease called tick-borne spotted fever in humans, and that's been described in Europe and in South America, but there hadn't been any reports from that end, the eastern end of the Mediterranean, so-called Levant, up until then. So we're not quite sure what that means, but this is surveillance. We're just looking to see what's out there and what is the potential in the future. Assembly 2, we found that there were these closest genome sequence relatives belong to species known as Rickettsia africae and parkeri, but those closest genomes were over tens of thousands of SNPs away. So what that meant was that we weren't looking at a genome from the same species. What we did then was we did look inside our genome and look at some signature genes, including 16S, but some others as well, and we looked in the databases for close matches to those signature genes, and we did indeed find those. They were matches to a species that had been named Rickettsia barbarii, and so what we'd actually turned up was the first genome sequence in this species. So although the species had been described from signature genes alone, it hadn't had a genome yet, and we provided it. We don't know whether this is capable of human infection. It hasn't yet been described associated with human infection, but we do know that it occurs in human parasitizing ticks. And so, again, this is surveillance. We're looking at what is the potential out there in the natural world for causing disease in humans. When we looked at the dog tick metagenomes, we did find something rather surprising. And this illustrates a point which is a powerful point about shotgun metagenomics, which is that it's open-ended. You're not restricted in what you find. You know, DNA from anything that's in there will turn up if you're using appropriate methods for searching and analysing your DNA. And what we found was we got 11-fold coverage of canine parvovirus genome in a dog tick from the town of Tubas. When we did blast comparisons, we showed the closest related to that was a strain from China, which again suggests, as we've said before, pathogens don't bother with passports. They don't honour international boundaries. They spread around the world pretty quickly and move easily. To provide context for this, canine parvovirus enteritis is a serious issue among dogs across the world. It causes disease and death among dogs. It emerged in the mid-1970s, and it's thought that the usual mode of transmission is fecal-oral. So one dog gives it to another in its feces. Dogs eat each other's feces or sniff them and whatever. But we do know that when the virus takes hold in the dog, it produces a marked viremia. It spreads in the blood, and you get large amounts of virus in the blood. And also, it had been shown in the lab that hard ticks could actually take up the virus, and it persisted in hard ticks in laboratory conditions for periods of time. So what we found here was the first evidence, just using metagenomics, that ticks can carry this virus in a natural setting. And it raises the interesting possibility that ticks might contribute to the spread of cane and parvovirus among dogs in Palestine and perhaps more widely as a neglected potential route. Another thing we found were what we call endosymbionts, which are bacteria which live within the ticks and are necessary for the ticks to survive, create essential nutrients that the ticks need to survive. One of the weird things about the way in which this works in ticks is that many of those endosymbionts are actually close relatives of pathogens that can cause disease in humans and animals. So Coxiella is best known as the cause of a disease called Q fever in humans, but in the ticks it appears that it's become an endosymbiont. And we found, again, a couple of assemblies here that we recovered, one from a sheep tick, one from a dog tick, and we showed that they were very similar to other similar strains that had been recovered from the area. So both recovered from Israel, which obviously is not far from the Palestinian West Bank, and suggesting that perhaps we are recapitulating or recovering something of the population structure of the ticks, and that the ticks show a geographically restricted population structure. But this is still very early days. These kind of analyses are have not been done very widely, and you know, whether we will recover the same endosymbionts from ticks all around the world is an open question, an interesting one. So what we found using metagenomics, that we could confirm the tick and the host DNA identification all in the same process. And we know that, you know, the morphological criteria are hard. We also got genome resolved characterization of key bacteria, pathogens and endosymbionts. And the key point is that metagenomics finds the unexpected. So if you devise a diagnostic test for a particular pathogen with some PCR primers specific to that pathogen, that's all it will tell you is the pathogen's there or not. It won't tell you whether there's something else there that you didn't expect, whereas the open-endedness of shotgun metagenomics, it does allow you to find the unexpected. Okay, any comments or questions on that particular tapper? No? It's been written up. The paper is available there. I'll send you the slides. I've got the link in if you're interested to see it later. The next case study is looking at the gut microbiota in ICU patients. That's patients in the intensive care unit, sometimes called the ITU, or intensive therapy unit. These patients are the sickest patients in the hospital, and they have a wide range of needs. They usually need ventilation, mechanical ventilation when they're in the intensive care unit. There's a high use of antibiotics in that setting, and these patients are prone to infection. The other reason they're prone to infection, apart from their being ventilated, is that they have intravenous lines which give deep access to deep tissues for bacteria that are on their skin. And so they often do get infected. And we know there's a crucial role of the gut microbiota in this. The gut microbiota acts as a reservoir of potential pathogens and for antimicrobial resistance in this setting. That was already known before we did our study. And it was, there was some evidence to suggest that low microbial diversity is linked to adverse clinical outcomes in this setting, and it's generally seen as a bad thing. If you have a simple gut microbiome rather than the highly complex one that healthy people have, that's considered a bad thing. So we decided to look at what happens to this gut microbiota in ICU patients. How does it respond to antibiotics? Do the antibiotics select for pathogens? And we were curious to see what we could discover in this setting simply using metagenomics without having to do culture as well. And at the back of our minds was this question that could we do fecal microbiota transplants in intensive care unit patients and actually provide them with increased resilience against infection and improve their recovery. Some of you already know about fecal microbiota transplants. This is a kind of exciting new therapeutic modality it's quite disgusting when you think about it. It's basically feeding shit to patients as a way of restoring the diversity in their gut microbiome. So those are the things in our mind when we started the project. And this was a project that was a collaboration with a clinical microbiologist, one of the ICU physicians, and 3 of us who were interested in microbial ecology and genomics. So what we did was we as a team, not me, we collected samples from 30 intensive care unit patients at the Queen Elizabeth Hospital in Birmingham, collected the relevant clinical metadata that went with it, and they were all subjected to shotgun metagenome sequencing on the Illumina NextSeq. And then we went in and we did a taxonomic classification, antibiotic resistance determination, and we did some strain level resolution of what was in there in a crude way, not in the sophisticated Chris Quince way, but nonetheless we did find some evidence of strains in there. And to cut a long story short, what did we find? We found that 2/3 of these patients experienced extremely low gut microbial diversity, and 1/3 of them really ultra low diversity, where in those patients a single organism had taken over their gut microbiome. So in healthy individuals, kind of people in this room, we would expect to document hundreds of different bacteria in your gut. Whereas in these patients, 90%, 80, 90% or more of the biomass in their gut was a single organism. So another way of putting that is in about 3/4 of the patients, we saw episodes of gut domination by pathogenic strains. And you can see here listed the patients, their age and sex, the clinical features that they had, and the peak pathogens that we saw, and the percentage abundance. So even the top one there, 17% of the gut microbiome being made of a single organism, Klebsiella pneumoniae, is pretty disturbing. But when you get down to the next, E. coli, 80%. We get down to Enterococcus faecium in one case, 99% of the sequence reads coming out of the patient were that organism. I was actually blown away by this. I kind of expected there would be perturbations in the gut microbiome and that we might pick up pathogens, a few percent here and there. But I wasn't prepared for the idea that the gut microbiome would effectively be replaced by a monoculture. So again, this is something that doing shotgun metagenomics reveals very interesting findings. One other thing that we found was that in 3 patients we recovered Enterococcus faecium genomes as mags from the metagenome, and they were quite closely related, only a few dozen SNPs apart. And they were in adjacent beds, in adjacent rooms in the intensive care unit at the same time. Because they were dozens of SNPs apart rather than just a handful, it's hard to say with certainty that there was direct patient-to-patient transmission in that environment. But it certainly hints at the idea that there is transmission, what we call nosocomial transmission. There is transmission within the hospital of a hospital clone that is lurking in the hospital and maybe in the environment, maybe in the gut of staff or other patients, that is moving around in that environment. And again, this is an insight that just came out of metagenomics. Turn the handle, do your metagenome analysis, and you find interesting stuff. This just shows you the picture graphically of what was going on in some of those genomes. You can see that we get a consistent picture when we take samples from the same patient, serial samples. We see an interpretable picture that they tend to have the same organism. It goes up and down. Some cases it goes away completely. In some cases it goes, as you can see in the bottom 153, it goes away to almost disappearing and then comes back again. And so this is, it's clear that the gut microbiome is actually being damaged by their time in ICU and particularly by the broad spectrum antibiotics they're getting. What we found when we tried to look at statistical links with the patient's clinical conditions, we were a bit disappointed that we didn't find any obvious thing that the severity of their illness or the consistency of their stools or anything like that had a link. But what we did find was that there was a link to meropenem. and this is a very broad spectrum antibiotic. And we found that there was a significant link there to the ongoing use of meropenem that did clearly lead to decimation of their gut microbiota and domination by a single pathogen. And that provides you a segue into what you're going to learn next week, in that you're going to learn a little bit about how we might understand how meropenem works in E. coli and how resistance might emerge. We did also document some other things. We noticed that some beneficial commensals were lost in most patients. And so the idea, the question is, what happens when you do these terrible things to the gut microbiome? It looks in this setting as if this gut microbiome is actually very fragile. And things that are normally thought of as the good bacteria just get lost. Whether they ever come back, whether there is recovery, is an interesting question. It's not one that we could actually document because we didn't follow the patients up after they left the ICU, and we don't know. There was a study done on, I think it was giving meropenem to volunteers around the same time, just after we did this. And they showed that the volunteers' gut microbiome was still not quite back to normal 3 months after a single dose. So it's an interesting question that arises. How easy is it to destroy? Is there, when you take antibiotics, do you inevitably lose things from your microbiome that you do not gain back again? Gut domination by commensals. We saw that sometimes it wasn't pathogens; it was things like Methanobrevibacter, which is an archaean, come back, and we had to leave the question dangling as to whether maybe if you restore diversity by giving fecal microbiota transplants, you might do some good in this setting. It's very messy doing clinical research in these kind of very heterogeneous patient populations. There is work going on with patients receiving stem cell bone marrow transplants where you get rather tighter results, and the rationale seems to be a bit clearer that this would be a good thing. But nonetheless, it's certainly something that should be entertained and investigated as we go forward. So in conclusion there, that paper's been written up, and we've shown that, shotgun metagenomics does provide interesting information. It does give us evidence of these clonal blooms of multi-drug resistance pathogens. And we've got insights into the pathogen biology and epidemiology. We could actually identify the strains, the sequence types of the things we were finding. So it was really quite interesting to get that far. Right, the last few minutes, I'm just going to tell you a little bit about chickens. Now, why on earth would you want to study the gut microbiome of chickens? Do you need a reason? Well, there are plenty of reasons to study chickens. It's the most common domesticated animal on the planet. It's the most common bird. It's the most common dinosaur on the planet, because birds are dinosaurs, don't forget. It accounts for a considerable biomass, proportion of the biomass. So chickens outweigh all wild birds combined on this planet in terms of biomass. It's the most important food production animal in the world. It's also played a key role in the history of science. Aristotle described chick embryos. Darwin's account of domestication focused on chickens as well. And it's a laboratory organism for research in many fields. But of course, we don't need a reason. The only reason we need is to be curious. Curiosity-driven research is one of the highest achievements of human civilization in its own right. So we don't need a reason. So what we did was we took chicken shit. We actually engaged in something that, I don't know if you come across this term, culturomics. It's a slightly ugly neologism from Didier Raoul's group, where he said that actually you don't have to do everything by metagenomics. You can culture stuff, and if you do that high throughput, do it well enough, you can actually get better information sometimes from a sample than you can from metagenomics. It's kind of making the point there. So we did that. We also did metagenome sequencing, and then we did reference-based phylogenetics using Kraken, and we looked at reference-free approaches where we created MAGs as well. So this was work that was done in collaboration with people here, Evelyn and Falk, two other group leaders here in in the quadrum. Rachel Gilroy, shown there as the postdoc that did most of the work in my group. And Roberto Laraggione was our collaborator at the University of Surrey. And he was the one that provided us with the material from chickens in the first place. And this very rather busy slide here just shows you the flow chart of the work we did. We basically we use Kraken and MegaHit and MetaBat and so forth, all commonly used things. We used a GTB toolkit at the end, and we found lots of novel species and lots of known species as well. So what we did was we, in addition to looking at a couple of dozen chickens from Surrey, we retrieved all the publicly available chicken gut metagenomic data sets that were in the databases. So that gave us over 600 samples from 10 different studies, 12 different countries. And we ended up then analysing all of those. And we generated over 5,000 mags. And we grouped them into metagenomic species. We found 846 clusters when we did that, spanning a range of different phyla, lots of novelty there, and we found over 340 novel species there. So this is the obligatory phylogenetic tree that you show of all the work you've found, all the things you've found in your study. And you can see the Firmicutes were predominant there, but they weren't the only phylum. We did find several other phyla represented in this environment. One of the interesting things that we did do, I think was fairly novel, was that rather than just say we're going to call our mag 123 or mag, ABC 123, which is common practice across the field, we actually said no, these are new species. They need to have new names, user-friendly Latin names, and so we actually created over 6,000 well-formed Latin binomials for these, and because the taxonomic experts say that it's not good enough to put this in supplementary data, we had to put it right in the body of the manuscript. So we ended up with a paper that was hundreds of pages long, and we had to pay an extra charge to publish such a long paper. It probably is the largest publication of Latin binomials in a single paper ever. Probably, I don't know for certain. And in fact, as a spin-off of that, we then, working with a nomenclature expert at Haranorum, we created 1 million new names that could be used off the shelf for bacteria going forward. Because one of the key things that's happening with metagenomics is we are discovering new species in abundance. And we're clearly at the stage, I think we've said this earlier in the week, we're at the stage already where most of the species that are known to us are known from metagenomic analyses. They have not been cultured. And that is only going to get worse or better, whichever way you look at it. If you're a fan of metagenomics, it's going to get better. Culture, what a silly 19th century approach. We did actually do culture though, and we got 282 isolates, and of these, we were surprised that chicken shit, as I've explained to you, it's a common setting. Across the planet, there are chickens everywhere, but nobody's looked in detail at it, and we found 30 new species in these chickens. Interestingly, we found a very close relative of E. coli in there, which had already been sort of delineated as a potential new species, but just given an alphanumeric code name. And we actually said, well, we've got the isolate, we can now deposit it in the data banks, and we can give it a new name. So we named it after a guy called Tom Whittam, who was an early pioneer in the population genetics and genomics of Escherichia and Escherichia coli, who sadly died quite young, 10 or 15 years ago. So again, that paper has now been published. If you're interested, you can look at it more. In fact, I'm just thinking about it, you all looked at it already because we said it as part of the aptitude test. I sent that paper as part of an aptitude test to the AI, and the AI did a good job in summarising the paper and producing a press release. So we have to think next year about how carefully how we do these things, but interesting. So that's just a few vignettes of what we've been doing, and it's just to. I mean, Chris already gave you some very powerful examples of the versatility and power of metagenomic analyses. I've just added a few more. We're actually continuing to do this. We're studying horse guts, pig guts. We've had a paper on pig guts already. Zoo animals. We've got a big study where we're just been collecting fecal samples from zoo animals and just sequencing it. Again, simply with the idea of discovery in mind. Let's just find out what's there, and we will find hundreds, probably thousands of new species, very simply, like by this kind of approach. We've also been doing some functional profiling of the chicken gut microbiota, looking at how it was. If you give the chicken gut microbiota starch, we're looking to see what happens, which organisms increase in abundance and benefit in that situation. Another area that we've been looking at is looking at the healthy human esophageal microbiome. And again, you think this is a fairly common setting. We've all got an esophagus. Why has nobody looked here before? But there's been hardly any work done in this setting, and certainly almost none looking at MAGs. There was one previous paper. And the other thing I'm spending a lot of my time doing is just working out how we're going to give names to all these millions of new species that we're going to discover in the coming years, decades, centuries to come. And if you're interested, there's some of the papers we've published in these related areas. And that's it. It's just a brief overview of the applications of metagenomics from our own experience. So has anyone got any queries or questions? How do I stop recording?